Welcome to Swastome, modern solutions for healthcare, built for India's tomorrow, shaping the future today.
My privilege to introduce our esteemed speaker, Dr. Utso Guha Roy. Dr. Roy is an accomplished consultant pathologist based in Kolkata, India, holding an MBBS and MD, with over 7 years of extensive experience in clinical diagnostics. His core expertise lies in cytology and histopathology, where he has contributed significantly to accurate disease diagnosis and patient management. Beyond conventional pathology practice, Dr. Roy is actively engaged in the evolving field of digital pathology. He is particularly interested in the integration of both generative and non-generative artificial intelligence into diagnostic workflows, working at the intersection of medicine and advanced computational technologies. He is also involved in AI-assisted laboratory medicine research and has contributed to cancer screening initiatives, helping bridge the gap between technological innovation and real-world clinical application. In addition, Dr. Roy has authored multiple peer-reviewed publications, reflecting his commitment to advancing scientific knowledge and improving diagnostic precision. We are truly fortunate to learn from his experience at the forefront of modern pathology and AI-driven healthcare. Please join me in warmly welcoming Dr. Utso Guha Roy.
So good evening, everyone. Thank you for joining today, and thank you for that nice introduction. I'm Dr. Utso Guha Roy, a consultant pathologist and increasingly an AI educator. So in the next 20 odd minutes, we will be talking about something that is a very significant change in the diagnostic field that has happened in the recent time. So that is digital pathology and AI augmented future diagnostics. So before we talk about solutions, we need to understand that what is the problem. So it's not a small one. So India have recently crossed 1.4 billion people, and that's not just a demographic milestone. So it's a healthcare stress that's happening in real time. But the challenge is not only the volume, so it's a complexity. So we are in between a developing and a developed nation. So we have the burden of both the non-communicable diseases as well as the infectious diseases like malaria, HIV, AIDS. So they haven't gone away, and now we are also seeing an explosion of the cancers and the non-communicable diseases like diabetes and the cardiovascular risks. Adding to this, there is a tropical climate, there is infrastructure gap, there is a cold chain vulnerabilities, and so you have a system that is under extraordinary multi-directional pressure all the time. So that is the reality of our diagnostic service that we are undergoing every day. So for example, let's meet Dr. Sharma. She's not a person, so she represents every one of us as a diagnostic field. So one pathologist in India caters to around 65,000 citizens, and which is far below the global standards. And the major labs around, processes thousands of slides per day. That is 1.2 million slides per year. So that phrase I keep coming back is we need to unchain the expertise from the physical location. So Dr. Sharma may be brilliant, but that brilliance has geographic problem. So digital pathology and AI can fix that. So here's a number that we should see. That's around 100 to 150 slides one person has to see per day or shift. So it could be hematology, it could be cytology, it could be histopathology. So in a high income countries like the West, this workload could trigger serious concerns, but here it's just normal Monday or Tuesday. So, but when we push humans past a sustainable, limits, the cascade is predictable. So diagnostic fatigue sets in, errors rate climbs, backlog grows, and eventually talented professionals, leave the field or become less active in the field, which makes the shortage, and that makes workload more for those who are in the system. So it's not a criticism for our colleagues, it's a systems problem, and a systems problem needs a systematic solution. So what could be the solution? So not working harder. We have been working harder, so we need to work smart with the advent of AI and the technology that has happened in the last few years. So the AI doesn't get tired at a slide 80 or slide 1000. It doesn't lose focus after even a long night shift, and it can handle repetitive high volume tasks like cell counting, malaria screening, so flagging the normal slide, so that Dr. Sharma and all others can focus their expertise on the cases that genuinely need them. And apart from that, we can also adopt the hub and spoke model that we always tell about. So like centralizing specialist expertise in major centers, while the samples, not the actual slides, maybe the digital whole slides are collected and sent to the central area from the peripheral facilities. So this is what digital pathology can enable us right now. So we can call digital pathology as a third revolution in the pathology domain or the laboratory medicine domain. So the first revolution was something like a IHC or immunohistochemistry, as we call, that is detecting specific proteins in the tumor cells. Second revolution could be the molecular diagnostics, where we could read the genome. And the third and ongoing one is the digital pathology and computational pathology model. So where we are turning entire tissue into image and into computational data. So we have to remember that each revolution didn't replace the pathologist or the laboratory medicine professionals. So it actually made them have some added superpowers, added powers to diagnose patients better way. So this revolution will be no different. So it will add another superpower in our bucket. So coming to whole slide imaging or WSI, that is we call. So this is the foundation of everything that we are discussing. Can go to full screen also. So it is the foundation of everything we are discussing. A scanner captures the entire glass slide at gigapixel resolution. So these are not just photo micrographs we sometimes take from the with our phone or with some adapter with the microscope. We're talking about files that are gigapixel size, gigabyte sizes, and each one is a complete navigable diagnostic quality of image. So the implications are obviously profound. There is no more shipping of glass that can break during shipping. There's no more waiting. It could be transferred just on real time. There is no degradation of stain with time, because when you keep the slides in long time for in a boxes, the stains become hazy. And second opinions become more frictionless, and subspecialty expertise becomes more accessible. And this isn't just better microscope, it's a liberation from the physical location. So wow, if you look inside a digital scanner, how does it actually work? The scanner does three things brilliantly. One is high-speed capture at multiple magnification. Next is automated stitching, that is thousands of image tiles are seamlessly joined into one cohesive whole image. And critically, another important thing is, for example, those tissue sections where it has more than one layer, or in cytology where we know there are cells are overlapping. So in those cases, we have an important thing called jet stacking. So in jet stacking mode, they capture the images at multiple focal lengths and to simulate the fine focus adjustment of our microscope that we do when we are adjusting to look in the those areas. So importantly, the scanners automate the mechanical part of the microscopy, stage movement, focus adjustment, image capture, and ultimately the result is a digital asset that is many ways richer than an original glass slide. But I want to be very transparent about something, that this transformation is as much as an IT project as a clinical one. So these WSI files are pretty big. So maybe 4 GBs, 5 GBs each. So you need a tired storage approach. So it could be a hot storage with very high speed. So which capabilities for the actively reporting ones. So those are ongoing reporting. There could be warm storage for recent archives and some for cold storage for long-term archiving. And also we need high bandwidth networks, because these images has to be transferred from the scanner to the LIS and to the, again back to the storage system, maybe cloud, maybe SD. So that means we need a very good bandwidth of internet. So this is also an IT project as well as a clinical transformation. So it will require planning, investment, and genuine institutional commitment. So going ahead, eyes open is essential. Another issue that we face sometimes, because it's a practical headache. So every scanner vendor has their own proprietary format. So it could be SVS, it could be NDPI, it could be TIFF. So this creates a real interoperability problem, because ultimately we want to scan at one site and report at another site, or we want to use that scanned image for some AI analysis. If there is not flexibility, then the whole purpose is gone. So the solution is what we call the DICOM, that is the digital imaging and communications in medicine. So this is an universal standard for medical imaging, and it has been well adopted in radiology for many years. So it has to be also asked from the vendors that your image output should be DICOM compatible. So ultimately, then only we can use the digital pathology sustainably and scalably, and it protects your lab from technologically isolation. So now that we are digital, so we can introduce our new partner, that is artificial intelligence. Think of it as not as a replacement of the pathologist. It is more like a co-pilot, not an autopilot. So basically, we have to understand that for any AI model, may it be in healthcare or in other sector, is as good as is the data it learns from. So this is the critical garbage in, garbage out theory we say. So in medical AI, it's not just a technical concern, it's a patient safety concern. And building high quality training data set requires expert pathologists to manually annotate the images, mark the cancer here, normal tissue here. And this is actually times intensive, expensive, and complicated by the fact that sometimes, expert pathologists may have some disagreement about some of the areas. So this inter-observer variability is not a failure. It reflects the actual complexity of the tissue architecture and the tissue interpretation. So the Indian pathologists who are participating in these annotation projects are actually not doing any administrative work. They are actually building the foundation of the next generation of diagnostics. Quick conceptual distinction that is worth understanding is the traditional machine learning requires human to define the features the algorithm should look for. Say it could be roundness of the nucleus, it could be texture of the glands, and so on. So humans had to articulate the rules. But now with the advent of deep learning, so the situation is kindly reversed. So you have to feed the neural networks thousands of labeled images. So you give 10,000 tumor and 10,000 normal images of a tissue. It will on its own self discover the relevant patterns, and maybe sometimes even patterns that are invisible to the human eyes are very subtle. So it's just mathematics at scale, not magic. So it is actually genuinely transformative for histopathology, where tissue patterns are extraordinarily complex. So how does a machine actually see? So it uses a process called computer vision. So the workflow has four basic steps. The first is acquisition and preprocessing. So where the cleaning of the data, the cleaning of the image, and stain normalization is done. Second is actually the segmentation. So we have to separate the tissue from the background. And next will be the feature extraction. That is the extraction of the vital details from the image. So it could be nucleus, it could be glands, it could be vessels, it could be cells, anything that is of importance for the diagnosis or for the training. And ultimately the quantification. So everything has to be converted into objective mathematical measurements. So ultimately this is remarkable because traditionally pathology is kind of a qualitative discipline. So it becomes now a quantitative one. Numbers which are reproducible and comparable across institutions and at different times also. So two basic things of computer vision, the CNNs, they are the actual workforce behind most of these models. So that is the convolutional neural networks. So how they do actually, it processes the scanned image, the gigapixel images through layers of mathematical filters. So one could be detecting the edges, one could detect the patterns, one could detect the shapes. So the early layers generally see simpler structure, while the deeper layers recognize higher level of features like tumor architecture and tumor microenvironment, etc. So that CNNs can perform classification like benign to malignant, or they can also help in segmentation, or they can also identify mitotic figures. So in recent studies, we are seeing that in several of these tasks, the CNN performance is almost matching with an expert pathologist. So standard CNNs are very good at seeing pixels, but they treat our slides like a flat, rigid grid. So, but in reality, histopathology is like a topology. So it's not just where a cell sits on an X and Y axis, it also has a dimension. So here enters the role of graph neural networks or GNNs. So think of it as like a social network. So where we treat the nodes, the cells as individual nodes and the spatial interactions as edges. So this is a game changer for things like TME mappings, like the tumor microenvironment mappings, where we need to quantify exactly how the tumor infiltrating lymphocytes are interacting with the tumor, because now we have something called the immunotherapy, and which is very good, proving to be very good for many of the cancers. So ultimately, the CNN, we can see like CNN is looking at a cell's face like a selfie, whereas the GNN is looking like a contact list. So ultimately, because in cancer, who you are hang out with will determine the prognosis of that patient or that tumor. So one of the biggest advantages of AI is that its ability to quantify what we traditionally estimate visually, like morphometry. So we can precisely get the nucleus size and shape measurement. We can do biomarker scoring like HER2/neu, ERPR, Ki67, PDL1. All this can be standardized and reproducible. And another vital aspect I was talking about is the spatial analysis, which helps us to identify tumor to immune cell response. So this actually reduces the subjectivity and improves our inter-observer variability and strengthens the clinical confidence. So for research limited settings, this reproducibility is essentially very valuable. It helps us to ensure that the diagnostic quality does not vary between center to center. So there are many tools that are available, but very few of them are FDA cleared. For example, the Page Full Focus, the Hello AP, the Path AI site, and there is one tool called the. These are all soft hardwares on the top platforms, and for software aspect, there is IBEX Prostate. So it's a FDA 510 cleared, but it is for prostate cancer in core biopsies. So we have to be very sure that these are actually approved for specific tumors for specific criteria. So it is not approved for breast. It is not approved for prostate, maybe whole specimen. It is approved for prostate core biopsy only. So we have to know which areas it has been approved and then use them accordingly. A few other use cases of AI could be digital hematology. So stepping into hematology, like this is enormously relevant because we know the disease burden we face. We have the malaria, we have the cancers. So, but many of these functions are now available in the digital, what we call the automated hematolyzers. So they are 5 part, 6 part, one the top end models have flags for blasts, have flags for NRBCs, have flags for malaria also. But AI can also be used to screen millions of red blood cells for morphology, abnormalities, to find out WBC differential, find out blast, find out malaria. A few companies in India are also doing good job, like SIGTuple. They have a model called, I think, AI 1000 or AI 100. So there's a scope for AI in also digital hematology. Coming to another important field is the flow cytometry, that is has a immense role in leukemia or the blood cancer. So flow cytometry has always been a kind of a subjective element. This is the what we call the art of getting, and the art depends heavily on the experience and expertise of the doctor that he is doing. But AI replaces this subjectivity and with dimensionality reduction algorithms like TSNE, UMAP, and it can also reveal cell clusters traditional methods may miss entirely. So this is where another aspect is coming on, and in minimal residual disease, where finding even one leukemic cell in 10,000 is very important. So AI can offer a superior sensitivity in that area, because ultimately we have to know whether the patient is actually in remission or not. Coming to genomics to some extent, so karyotyping, as we all know, was a very manual, laborious task for the technician or doctor who is doing it. So he has to literally cut and paste those images. But now this can be done automated way by the AI. So it will segment and arrange the chromosomes in minutes rather than hours. Also in fish signal counting for maybe HER2/neu or ALK rearrangements. So this ultimately helps in a faster turnaround time. So it could be rapid prenatal screening for aneuploidy. It could be cancer diagnostics. So getting a faster turnaround time, getting a report maybe days ahead changes a lot in the output of the patient treatment. And another area is the next generation sequencing or the NGS. So NGS actually produces a huge amount of data or big data, which is very difficult to analyze humanly. So AI makes it tractable For rare diseases diagnosis, AI can prioritize the variants of unknown significance by cross-referencing them with their huge computing power and with referencing genomic data and patient phenotype. For oncology also, it can detect the ultra-low frequency somatic mutations in the liquid biopsies event, thus enabling early cancer detection from even a blood draw, not only from a tissue. So we are currently moving like steadily towards a world where the patient's prognosis and optimal treatment can be predicted from a combination of these tissue images or their genomic profile. So this brings us to this slide, that is what I consider is the most exciting horizon. So pathology traditionally was one question, what is this? And we got a diagnosis. But now with the advent of AI and digital pathology, we get two more asked questions, that is what will happen and how the treatment will work best for which person. So here comes the role for multimodal AI. So we can combine the whole slide imaging, the data from the genomics, the data from the EHR data, the patient data, and get a actual precision medicine, not something hype, but actual data driven precision medicine, which is the ultimate goal for the future. But we have to keep in mind the ethics, privacy, obviously, because everything is not very great. So we have to have an explainability models. The models have to have explainable, because why did the AI make a diagnosis? It is a benign or a malignant. If a clinician cannot understand, if a pathologist cannot understand, he won't be able to trust the system. So that models has to have an explainability mode built in, and it should be obviously free from biases. Like data model trained on Western data set may not work as well in the South Asian population or Indian population. And thirdly, the data protection like the HIPAA, GDPR. Now we have the DPDP Act in India also. So the patient consent frameworks must be respected when training models on patient images or patient data. So the trust in AI is earned through transparency and fairness, and we should hold that for every tool to that standard. And obviously, we have to maintain, navigate the legal landscape. So in US, the whole slide imaging system are the class 2 medical devices, and they require 510K FDA approval. In our regions, we have to adhere to the guidelines from the bodies like CAP, NABL or NABH. And there are also implications for telepathology. So we must understand the liability and the licensing frameworks as per the local regulations. And ultimately, the compliance to the local regulators should be mandatory. So here's the vision I want you to carry forward. So 3 outcomes that AI and digital pathology can unlock together. One is democratization. So the specialist expertise can reach the remote and underserved population. Second is efficacy. So we have a faster turnaround time, fewer bottleneck, and we have a higher accuracy. So not AI alone or not humans alone, but the combination will achieve results neither could reach independently. So digital pathology is pathology evolving into a data science, and that is not a threat to our profession. This is our profession in an upgraded way. So this is my last slide. I will leave you with this thought that the future of diagnostics will not be decided by technology alone, but by how wisely, how ethically, and how inclusively we use it. The tools are here. The evidence is accumulating by multiple studies that are ongoing. The patients, whether in Mumbai or in a rural district hospital, they are waiting. So thank you all, and I'm happy to take questions.
Hi, doctor. India is lacking digital scanner. Very few hospitals are actually having scanner. How do you think without digital scanner, digital pathology will move in India?
Yeah, I actually face the same issue because to be frank, in Kolkata also, very few setups, maybe only Tata has a digital scanner. So yeah, that is a big issue. But the thing is that horizon is changing. So the Leica, maybe the Philips and the Hamamatsu, they have a very costly ones. But in India, nowadays, Morphil and other companies are making it with lesser cost, maybe around, I'm not quoting the actual price, maybe it's around 25 lakhs, something around that baseline. And there are other companies I recently met with, they are also quoting the price around 10-15 lakhs. But yeah, still is a big ask. And it's something we have to change in our mindset also, that digital pathology is not only for reporting, it has multiple roles for telepathology, for AI use, for Hatinu scoring, everything can be used after only the digital scanner is used. So that thing we have to convince our lab management, because to them it's only an ROI. And I understand they think that we can obviously report from a glass slide as we have been doing for years. So yes, the field is changing. I hope in a few more years, more affordable ones will come, and just looking forward to that day.
Okay. Thank you. So next question is from Ronita. What kind of background or noise one can face with digital pathology image analysis?
Okay, so actually it could be multiple. So there could be some artifacts multiple times. Major issue with histopathology, and that differentiates with radiology, is radiology is a black and white kind of thing. So it's very easy for them to categorize them and annotate them. But for histopathology, we see it's a multiple kind of staining. So H&E has a multiple graded kind of staining, and that itself causes a issue. So if the stain normalization is not done across the data set, it could be a very difficult for a AI tool to analyze if the model has not been trained on the same kind of image. So stain normalization has to be done, and many times we see air bubbles coming inside the slide. There could be artifacts coming in the slide. There could be ink droplets. So those things has to be understood, and even those things can be annotated as artifacts when training the models. So when I personally worked with some companies in annotations, so they were asking us to annotate those artifacts also. There could be a strand of cotton strand in the slide. So we have to annotate those areas as an artifact or some group to be ignored, and that input can be put into the AI model during training.
Okay. Okay. So the next question is from Mansi. So hi Swastam and Dr. Utso. I am doing my masters in microbiology. I want to learn digital pathology. Is there any course where I can learn image analysis and digital pathology from basics and analysis?
There are not very one. So I actually did one for Metropolis. So they have a course on digital pathology. It's a 3 months course. There is on one another from Global Health Academy, I think, conducted by Dr. Veena. Maybe you can find her on LinkedIn. They have a course, and there is one reputed course, but little costly. That is actually Digital Pathology Association (DPA). So they have an online course for 3 months.
Okay. So the next is from Rajot Bush. Thanks for the lectures. Can you start a course on this? Please, I have done biotechnology. I took some courses, but the course contents were not good. So
Let's
there
see.
are lots of questions in the comment section. So I think we will move forward.